All right, advanced biology students, this is our last lecture focused on cellular structure and its functioning. We ended with our talk on lysosomes. We'll pick up here with things known as vesicles and vacuoles. Vesicles and vacuoles, they are larger versions. Vesicles are larger versions of things called vacuoles. They are membrane-bound sacs with varied functions. A plant cell or a fungal cell may have one or several vacuoles in it. Vacuoles themselves can be food vacuoles that are formed via phagocytosis. Phagocytosis is something that we will look at a little bit later. It's cellular eating. You also have contractile vacuoles that we'll look at. These are found in many freshwater protists. They're going to pump out excess water out of the cell. And then in plants, you have the central vacuole, which is going to hold organic compounds and water itself. So some review, there is the central vacuole again. If you look at the electron micrograph of a plant cell, here you can see the central vacuole, and it is surrounded by a membrane called the tonoplast. So those are all structures that we have looked at. Now something new. Cell energetic organelles. This would include both the mitochondria, which is the site of cellular respiration, and the chloroplast, which is found only in plants and blue-green algae, because it is chloroplast are the site of the process of photosynthesis. Mitochondria and chloroplast are not part of the endomembrane system. We also have peroxisomes, which are oxidative organelles, and we'll look at each of those things. So mitochondria themselves are found in nearly all eukaryotic cells. They have a smooth outer membrane and an inner membrane that is folded to form structures called cristae. The inner membrane creates two compartments with the folds. You're going to have the inner membrane space that's going to be found between the inner and outer membrane. And you have the area called the mitochondrial matrix. And the area of the mitochondrial matrix is going to be inside within the inner membrane. So the area that's within the inner membrane itself. It is that area where you have some metabolic steps of cell respiration that are going to be catalyzed in the mitochondrial matrix. Other reactions are going to take place on the inner membrane itself. Therefore, it would be the cristae. So the cristae is going to present a large surface area. That's why the inner membrane is folded to form those structures. The larger the surface area, the more room you have for a reaction. So the cristae is going to present a large surface area for enzymes that are going to help make or synthesize ATP. So here, if you look at the structure of the mitochondria, which is the site of cellular respiration, here you have the smooth outer membrane, and then here you can see the inner membrane and the inner membrane foldings called the cristae. The area between the inner and outer membrane is the intermembrane space, and this area in here is the mitochondrial matrix. Mitochondria do have free ribosomes. Mitochondria also have their own circular DNA, known as mitochondrial DNA, which is pretty cool. The chloroplast is a member of a family of organelles called the plastids. Chloroplasts contain a green pigment called chlorophyll, and when we get to photosynthesis, we'll study the various types of chlorophyll and the different pigments associated. The different colors associated with the different types of chlorophyll. Chloroplasts are found in the leaves and also in other green organs of plants and the blue-green algae. Chloroplast structure includes the thylakoids, which are membranous sacs, and then on the inside you have the stroma, which is the internal fluid. The chloroplast itself is also a membrane-bound structure. You have the inner membrane and the outer membrane. But it's right in here within the thylakoids. They look like stacks of pancakes or a stack of coins. That's where one type of reaction of photosynthesis will take place. And then in the stroma, which is the inside area found between the different stacks of thylakoids, that would be the stroma itself. That's where another reaction will take place. Basically, one little, if you think of it in terms of pancakes, one of those things is a thylakoid. A stack of them is called granum. The other structure, peroxisomes. Peroxisomes are specialized metabolic compartments bound by a single membrane. Peroxisomes are going to play a role in the degradation of fatty acids and amino acids and produce hydrogen peroxide. Therefore, we have the enzyme catalase that is going to help break that hydrogen peroxide down. The reason we need that is because hydrogen peroxide, the reason we need catalase is because hydrogen peroxide is a poison to the cells. What happens is you get the dissociation of poisons into these things called free radicals. And ultimately, free radicals can lead to diseases down the road, like cancer. The cell's cytoskeleton is a network of fibers that are going to extend throughout the cytoplasm. So cyto is cell, so we're looking at cytoskeleton, the cell's skeleton. And it's going to organize the cell structures and activities and help anchor many of those organelles in place. Molecularly, there are three basic things that come together to form the cytoskeleton itself. They are the microtubules, microfilaments, and things called intermediate filaments. So if you look structurally, here is what the cytoskeleton would look like. It is not like our skeleton set. Basically, you could have the cytoskeleton reshape itself. So it supports the cell and helps maintain its shape. It's going to interact with motor proteins to produce motility. It's going to serve as a monorail or transport system for vesicles as they move through the cytoskeleton. It will also help to regulate many biochemical activities. The extracellular components and cell connections. Now we're going beyond the cell membrane to look at the cell parts or cell parts associated external to the cell. Most cells synthesize and secrete materials that are external to the plasma membrane, and we will focus heavily on the plasma membrane in the next chapter and test on these two chapters together. These extracellular structures include the cell walls of plants, the extracellular matrix, abbreviated ECM, of animal cells, and things called intercellular junctions. Plant cell walls are extracellular structures that are not found in animal cells. It's going to protect the plant cell, help maintain its shape, and prevent the excessive uptake and loss of water. You know that the plant cell wall is composed of cellulose fibers that are going to be embedded in other polysaccharides and proteins. Plant cell walls may have multiple layers. We have the primary cell wall and a secondary cell wall. The primary cell wall is going to be thin and flexible. The secondary cell wall is only found in some cells, and it's added between the plasma membrane and that primary cell wall. Between the two, you have the middle lamella. The middle lamella is going to be a thin layer that's going to be between primary walls of adjacent cells. So you have a primary cell wall of one cell meeting the primary cell wall of another cell, and in between you have the space that we call the middle lamella. Now, because the cell walls are rigid structures, because they contain cellulose, it's hard for substances to pass through that cell wall. So we need to have channels there, and the channels are going to be called plasmodesmata. And the plasmodesmata are going to allow things to be transported between cells that are adjacent to each other. So if we look, here we have the central vacuole of one cell. Here's the central vacuole of another cell. Here we could see the middle lamella. So right here we have the primary cell wall of plant cell A. Here's the primary cell wall of plant cell B. So we're looking at a structure just like this. And then in between there, you can see you have little channels that are going between one cell to another called the plasmodesmata. The plasmodesmata are channels that are going to perforate cell walls and allow water and small solutes, those are substances that are able to be dissolved in water, and sometimes proteins and RNA, to be able to pass from one cell to the next. And there you can see a better electron micrograph of plasmodesmata extending between cells. In the animal cell, you have this thing called the extracellular matrix. The extracellular matrix, animal cells lack cell walls but are covered by an elaborate matrix of proteins, lipids, etc. The ECM is made up of glycoproteins and other macromolecules that are going to come together to complete this external structure that's going to function and support adhesion, movement, and regulation for the cells. So here you can see the extracellular fluid. There is a typical cell membrane, and in here would be inside the cell because you're within the cytoplasm region. So intracellular junctions are going to be extracellular features that are going to allow for neighboring cells and tissues, organs, or organ systems will often adhere, interact, and communicate through direct physical contact. Intracellular junctions are going to facilitate this contact with the cells. Animal cells also have things called tight junctions, desmosomes, and gap junctions. Tight junctions are going to be membranes of neighboring cells that are pressed together, preventing leakage of extracellular fluid. These are very typical of cells that would make up our stomach because we don't want the contents of the stomach to be leaking into our body cavity. Desmosomes are anchoring junctions. They're going to fasten cells together and allow them to form strong sheets. And then gap junctions are going to play a role in communication. So these are communicating junctions that provide cytoplasmic channels between adjacent animal cells. So here you can see how the tight junctions would look. There's a cartoon drawing what it looks like through an electron micrograph. Desmosomes, and then gap junctions. So ultimately, in conclusion, the living cells rely on the integration of structures and organelles in order to function. For example, a macrophage's ability to destroy a bacteria involves the whole cell coordinating components such as the cytoskeleton, lysosomes, and the plasma membrane. And there's your picture of a macrophage taking in bacteria cells. So I hope you have a better understanding of cell structure function. Please feel free to write down any questions and ask them in class. This concludes Chapter 4. Thank you very much, and thank you for watching.